through this in detail. Okay? Go right ahead.
All right. All right, so our experiment is the egg cooker. We're cooking an egg in zero gravity.
Cool.
Speak up loud.
Okay. So our experiment involves taking uncooked, prepackaged egg dishes from in this box and putting them in our egg cooker here. And then when they're done cooking, we take them out and put them into here. This device does have temperature sensors built into it. So if the external temperature on this box ever is above 113 degrees Fahrenheit, an alarm will sound and we'll know to take our hands off of it and hit the kill switch. Strap down, and we also, in case that the microcontroller fails or any electrical part fails, we do have a multimeter here with a temperature probe and a hole in front so that we can get a temperature reading with this as well.
How long does it take to cook?
Oh, yeah, you can pull that out there.
These are the containers that they will be inside.
So you'll do that here. You'll pour them in there.
Pour them in here and then seal them with this silicone paste. It'll be sealed shut.
Okay.
Designed to grow plants in space. Since we will be using water, ours is double contained. That is, we call this our Lexan box. It is completely waterproof, and the tube we have here for our power cord is completely sealed. You can see the white sealant. We will be controlling this experiment via Bluetooth with this tablet primarily, and if that were to fail, back up via a phone. We have apps for both of them that can run the experiment.
All of our recording will then be handled by 2 GoPros, so we can then pull the information later on after the actual experiment. Those are then controlled by 2 Wi-Fi remotes on this far right-hand side, so we won't have any physical interaction between the internal box.
It's all ratcheted down, as you can see.
Again, we have the same kill switches on the right-hand side. We have a master kill, which will kill both projects in the case of extreme emergency. We also have our hydrofuge kill box here, and then on the top we have a USB control for the actual hydrofuge inside the unit.
So our experiment is a attempt to create a system to study the effect of microgravity on lubrication of mechanical systems. And so each of these modules will spin the gears up for 5 seconds and then let them spin down under their own momentum. The speed at which they spin and the RPM will be recorded by the optical encoders we have here and sent to our Arduino board. And then the output will be displayed on our computer over there.
We can just kill that. And this will be outside the glove box. This is how we power the experiment every time we're in zero gravity.
And there's a 5 amp fuse connected here to the battery.
In high school, I'm Cynthia.
I'm Patrick.
I'm Soleil.
I'm Jake Dunn.
So we're looking at surface tension and the effects that it has on crystallization in a microgravity environment. And here we have our acrylic nano lab, which is holding our acrylic crystal chamber, which is then holding our triple contained acrylic slides. And each channel slide has a unique shape to it that all holds a different surface tension that we're going to be testing. And this also holds our nesting board in our inner enclosure along with our piston. And we're going to be looking at the size, the shape, and the orientation and the distribution of the crystallized slides. And Patrick is going to tell you later of the nesting board.
And so, as you can see, we have a camera connected to our nesting board.
Nice
So,
loud. Speak up louder.
So this is our nesting board. It has a camera and a solenoid connected to it. And what it's programmed to do is when it's plugged into the computer, it will actuate the solenoid, puncturing the membrane we have right there. And then the lights will turn on and take a picture.
Sodium acetate solution, and sodium acetate is vinegar salt, and it's a food additive, so it's what makes sea salt and vinegar chips taste like sea salt and vinegar. And so that will be in there, and we're using that one because it crystallizes, it stays a liquid, and then once it's touched by something like an outside thing, it crystallizes almost instantaneously. And so it'll be triple contained. in the acrylic box, and then in the acrylic nanolab, and then within the glove box. And the other thing is that it crystallizes so quickly that if there were to be a leak, it would probably crystallize so that it wouldn't leak anywhere.
And it will be triple contained.
Yeah.
Those where you can put it in the water. Well, that's essentially the exact same thing. It's we have actually done it with that one in particular already, and it goes back, and then we can renucleate it and get crystals again. So, but for our experiment, we're only going to be crystallizing it during the zero gravity portion, and then we're going to be waiting, pulling it off, and looking at the slides underneath the microscope.
We're at Fairport High School. My name is Luke Dangler. I'm one of the flyers, and we're testing the effectiveness of UV radiation to kill bacteria in zero gravity. So our experiment involves us using petri dishes that are pre-cultured. They'll be cultured that morning of bioluminescent bacteria, and then we'll be exposing the petri dishes to a UV LED in this dark box and comparing the results of the control half of each petri dish that will be covered by an index card to the other half of the petri dish with a photo taken in our dark box. And then we have a computer program that will analyze pixel by pixel to tell us how much light is exactly being emitted by each half of the petri dish.
And School of Science and Math from Durham, North Carolina. And our project is a peristaltic pump. Right now it's currently inside the nano rack right there, as you can see, and it's a pretty self-running experiment. It's attached with a USB driver to the NESI board, and that runs outside to a computer that will be sitting on top of the glove box and just plug and run. Peristaltic pump, it's basically mimics the body's motion of like how you would swallow in order to pump a liquid through the system. And so we have a self-contained liquid combination of mineral oil and water that can be detected by a photoresistor, so to see the changes in net flow. And so that's all done with nitinol wires and a heating source, and that's inside the box.
Which will measure the resistance, so it'll get lower when it's pressed upon, and we can extrapolate force from that. We're also using pressure sensing film, which is in this package, to find the pressure points between the vertebrae.
How are you set up for that? You're just floating around, or are you sit down, or?
Yeah, so we'll be strapped, we'll be attached to the floor of the plane, and we have the paper contained. This will be a single unit once it's attached, and it will stay in the box until we take it out for the outreach items at the end.
All right. Well, my name's John Korber. I'm with Jackson Hole High School. Simply put, the goal of our experiment is to eventually manufacture parts in space. This is ours is in the box. The way we're doing it is inside of a nano box or nano lab experiment enclosure. We have a couple of different slide assemblies. Each slide assembly is a piece of metal, a small amount of magnetic wax held between two pieces of parchment paper, PLA plastic shelf inside of a 1/4-inch Lexan box. The power, we intend to melt the wax with Peltier plates. Which will allow it to conform to the shape of the magnets inside the magnetic array, inside the plastic case. We deactivate it, and then it hardens in microgravity. Post-flight, we'll be able to pull out this wax and examine the structure. The Peltier plates are operated with this switch box over here on the side.
So this is Counselor Oxal's experiment. We were inspired by two things. One was another high school. They had a hydroponic plant chamber that they grew plants in and siphoned off the rest of the water. And we were also inspired by the mission to Mars because we want to be able to eventually control the movement of liquids in zero gravity, because right now that's the hardest thing to do, we believe. So
Our eventual goal is to see if we can do things such as lubricating motors, growing plants' food, purifying air, things like that. So what we have here is a system that measures the rate of capillary action.
So we have our 2 nanolabs, they're duplicates of each other, where we have the Nessie board on one side and our experiment on the other. So in the experiment, you can see the plastic capillary tubes. That's where the water will be rising during the flight. The aluminum header on the side, on both, is acting as your finger when you're playing with the capillary tube. It's what will allow the capillary action not to begin only in 0G. We have our 2 cameras on top of the red mount, or inside of the red mount. They'll be recording our experiment, and as a backup, we have a GoPro at the base controlled by a remote to get additional footage if we need it. Capillary tube action. When they're in flight, they'll have a parabola to turn it on for 1 set. Then they'll do another set on the 3rd one. The flyers will go in, click the button, push down on the syringe to a set amount, and then turn it off. Then the next one, they do click, next parabola click, next one after that, other syringe.
I'm Eli Rand. We're from East Troy High School from Wisconsin. This is our experiment right here.
What high school?
East Troy, Wisconsin. Yeah? Okay. So we use RFID technology, which is radio frequency identification. And so we have a tag right here, and we're going to be testing how far away we can get it using just measuring the distances. And we hope to put on the ISS and have stickers that can go into tools. And then if the tools get too far away, then a light would flash, telling you that it's going to maybe hit a computer or get lost in a vent. So we have that, and just to keep track of tools so that they don't get too far away in 0 gravity.
We're from Tri-County Regional Vocational Technical High School in Franklin, Mass. This is Keith Boyle, Johnny Bello Vance, and Nathan Rogers.
So our project is a 0 gravity scale. What the purpose is to. All right. So the purpose is to find the mass of an unknown object in a 0 gravity environment. With this design that we have, everything is contained. So we have these weights that are bolted onto these force sensors.
That one doesn't have a weight on that side.
Well, this one doesn't have a weight on it, just to show you the force sensor. Everything is bolted on to the main axle. On this side, we do have a weight. It spins. So when it spins, it gets its own sense of gravity via centrifugal force. How we're powering it is just a simple USB connection from the laptop, and our laptop's going to be bolted onto the top of actually it's Velcroed on to the top of the glove box. It's just a 2-switch operation, so you don't need to have much human interaction. It's just flip a switch when you want data, flip a switch off when you are done taking data.
Tired yet? No,
Okay.
I'm excited. Let's go.
Okay, so what our experiment is, we're trying to see how gases and molecules break down in 0 gravity environment. So we have 2 hydrogen fuel cells right here. The first is used for electrolysis, which is used to, what we'll do is we'll soak a membrane, it's called a PEM membrane, and then we'll apply a current to it which separates the hydrogen, or the water into hydrogen and oxygen. And then we'll feed it to the second fuel cell to drive a circuit, which we'll be able to see the current and the voltage from. And that's pretty much the gist of our experiment. We're trying to, and it'll recombine and make water at the other end.
What quantities?
It produces 7 milliliters of hydrogen per minute, or it's like 0.6125 milligrams per minute.
The AM class of Governor's School ultimately wants to build a clean bot for the International Space Station to go around the inside. The first step in doing this was finding a proper adhesive material that we could use for this on our clean bot. So after searching, we came across something called Gecko Tape, which is based off biomimicry of an actual gecko. And for this test, we're going to be applying various amounts of forces and then finding the force to remove it. And so we do that with this load cell here that's hooked up to our computer. Everything is going to be Velcro down to the side or on top of the box as we have here. And this is the entire thing.
He mentioned how everything was going to be Velcro here. This is in between here
There's 2 foam plates, and they house the board that we're going to use. It's a fidget bridge, and basically all it does is that it hooks up to this load cell here, and it translates the data that it produces into the laptop so we can record it and make our data table. The 2 different foam plates are surrounded by a waterproof coating, and that is all taped together with the non-residue tape that they provided us. The wires here, which were exposed, are covered with electrical tape. That's to prevent any, possibly them snapping out or anybody, getting shocked, I guess. It's not really an issue anymore. The surface here that we're going to be pressing our Gecko Tape against was a piece of an old CD that we used. The edges are sanded down, and then they've been taped over with electrical tape. That's to prevent any sharp edges, anything that could potentially cut anybody else. And then there's a surface on the Gecko Tape, and that's more of an operational thing than it is more of a safety.
We're part of Cristo Rey Jesuit here in Houston, Texas, close to Hobby Airport. And as an experiment, as a testing of our experiment, we're here for the parabolic flight for the National Design Challenge by CASIS and NASA. And we have our experiment right here on this side of the board. And it's completely self-contained, no need for an external computer. And inside of it is an Arduino computer that will monitor the processes of mesoscopic lipids, which are just lipids that are, we're expecting to mimic the functions and the formations of lipids in microgravity. So we 3D printed lipids, of course, in plastic and magnetic shapes to see if they form as they should in microgravity. And we expect to do that by stimulating them with vibration motors that are also controlled by the Arduino. Vibration motors and electromagnets. Pushing both of them will do a soft kill, as well as pulling out the plug, a hard kill. Aside from that, it's self-contained, as I mentioned before.
We're from Clear Creek High School, and our experiment is focused on crystallization in microgravity. What we did was we designed a box in which we could contain all this, so we can have 3 layers of containment and safety and isolation from any chemicals at all times. We actually had some mixed tubes shipped in for us from nanoracks, which have been on the ISS and have had various fluids mixed in with them before. And the way we want to carry out this experiment is between every microgravity phase, every other microgravity phase, we will switch out these pairs of 3 mixed tubes. They slide out of this experiment testing rig right here, and they unclamp right here. I'm not going to unclamp them right now, but they unclamp pretty simply. You just pull this tab right here, and both chemicals come in contact and cause a chemical reaction. And we have these little, kind of like the glove ports that are on the NASA glove box. We have our own for where you can put your hand through and adjust various things on the experiment.
Yeah. For visual data. And we have 3 more down here, so we're going to interchange them in between the parabolas.
What were the chemicals?
Well, we have ethanol alcohol and egg white, which the ethanol denatures the egg white. And I forgot to explain this, but the expected outcome of growing crystals in microgravity is to have more uniformly shaped crystals, more geometric, because without the force of gravity acting upon the crystals, they can form more uniformly than they would on Earth.
Clear Springs High School from League City, Texas. We've got 2 experiments here in 2 separate groups, so I'll be explaining our first one. This experiment right here, we're going to be testing RCS engines and how they work, and we're going to be looking for the optimal angle at which we can get the most contact surface. We're currently just using liquids, but we're simulating the hypergolic fluids that will be used in space. So to start off, the liquid will start in these syringes. We're going to have a push button installed here in just a little bit that's going to turn the gears, push the syringes, it'll push it through, and after 6 parabolas, we're going to be changing each fluid, or each valve, to send it to the different injectors down here, and that's actually going to spray onto a sheet of absorbent paper. And in between, during the 2G portions, we'll actually be twisting this, which will shift us to a new sheet, so we'll be able to collect different spray patterns after each 0G parabola.
included in 2G.
Yeah.
Yes. Just the only work that will be done in 2G will actually be twisting this, just enough so that the new sheet comes along.
Still not an easy
project. We are separating air bubbles from water. This is to help deal with the problem of storing fuel or water in microgravity. Our first system is the thermal system. Basically, we are relying on heat differential. We'll have a heat pad on one end and an ice pack on the other. And basically, we'll be injecting air bubbles through the IV bag, and the air bubbles will be attracted towards the heat. From there, we have a bubble sensor. This will detect that there's a bubble, and then it will tell our solenoid and our syringe to extract that. The excess water will be traveled here. Our second system is we are using centrifugal. This is composed of VEX parts. We will be injecting water and air down these tubes. It will enter into the chamber from the very bottom. It's connected to the sides, and it will be injected from the bottom.
Right.
We'll turn on the chamber, and it will rotate. The water will kind of rush to the side. The air will stay in the center. From there, we'll extract it with our top syringe. All of these are bolted down and are secure. Everything is sealed. Our first level of containment is the experiment itself. These, our second will be this. It would be sealed with silicone all around.
and that kind of thing.
So could we put like absorbent tiles in here to? Yes, we could.
Yeah.
Not for thermal, because in order for the solenoid valve to open and the syringe to retract, we visually have to see this light here go from green to red, and that will tell us to push on the push button that will retract them manually.